NMR with nanodiscs, and ultimately to solid-state NMR with phospholipid liposomes. So what I'm showing you here are the two cartoons illustrating the sample types. A nanodisc is basically a patch of lipid bilayer membrane that is corralled by two copies of the apolipoprotein, this helical yellow string that happens to be the apolipoprotein that's also found in our blood. And these have become a very useful tool for solution NMR studies of membrane proteins popularized by Gerhard Wagner's laboratory. In the case of liposomes, we obviously have to go to solid-state NMR because these are now much larger protein lipid assemblies. And these are essentially shells of lipid bilayer membrane that encapsulates water and can be dispersed in water. So what's really nice to see in this case is if you compare the 3D, so the 2D slice from a 3D HNCA solution NMR spectrum taken in nanodiscs, and that's in red, with a 2D NCA solid-state NMR spectrum of the same protein in liposomes, you can see this very nice overlap of the resonances, suggesting that or indicating that the conformation of the protein in the two types of environment in the lipid bilayer is very, very similar. The nanodisc spectra were obtained at 45 degrees, the liposomes at 10 degrees, so you have slight differences due to temperature, but the lipid composition is the same, and the spectra really reflect that. And moreover, we took this further to proton-nitrogen correlation spectroscopy. So what I'm showing you here are a TROSY HSQC spectrum in red from the protein in nanodisc, which I believe was taken at 800 megahertz, overlaid with a CP HSQC solid-state NMR spectrum in black of the protein in liposomes, obtained at 900 megahertz with 60 kilohertz magic angle spinning spectroscopy. And again, you can see this dramatic overlap of the spectra, indicating that the structural, the conformation of the protein is maintained in both cases. And because we have a number of assignments from the solution NMR spectroscopy, much of it could be ported to the solid state. Now, in this case, we were very gratified to see that when we did the same ELISA assay with the protein in nanodisc, and actually even with liposomes, in this case we have coated vitronectin on the ELISA plate, now we have this very nice binding activity with the ale nanodisc compared to empty nanodisc, which have no activity. So in this case, we were able to show that the protein, at least the reconstituted protein, is able to bind to this blood vitronectin. And now the goal was really to go and map this interaction to understand it at the molecular level. Okay, so an aside, what is vitronectin? So vitronectin is this abundant protein that circulates in our blood at a level of about 500 microgram per mil. So it's highly abundant. It's important in a wide range of human processes that range from mediating cell adhesion through its interaction with integrins, shown right here, mediating coagulation and fibrinolysis through the interaction of plasminogen activators and plasminogen receptors, like PI1 and UPAR. It's important for mounting innate immunity against bacterial virulence factors, and that really happens through its interactions with the complement system, the system that's responsible for punching holes into invading bacterial membranes by forming these membrane attack complex channels that's shown here in the cartoon. It turns out it's also a major component of lipoproteins, things like LDLs. It's important for tissue culture and bioengineering applications. And most recently, it's being identified as a major component of the deposits, of the ectopic abnormal deposits that form in things like macular degeneration. And that's a whole other area that we've been interested in and are addressing right now, the ability to form these structures that are very interesting. So they're made up of a core of a lipid droplet surrounded by mineralized hydroxyapatite with a layer of yellow vitronectin on the outside. Very, very interesting structures. So despite its huge importance, there has been very little known about this protein from the standpoint of structure. And so we took it to heart to really try and produce regions of the proteins and characterize them structurally. The first thing that we did was to work with Greg Plano using bacteria that express, natively express ale, because these are Y-pestis bacteria, and trying to understand which regions of the protein of ale interact with vitronectin. So using these assays, and we published this in this paper in 2015, we showed that the presence of ale is absolutely essential for pulling down vitronectin from serum. Here's a control vitronectin, and here you see that when you have ale and normal human serum, you pull down abundant vitronectin. Whereas in the case where you don't have, where you inactivate the serum with heat, you have no vitronectin pull down. And in addition to that, we have embarked on a series of mutagenesis experiments which have shown that the extracellular loops of ale are absolutely critical for the interaction. So you can see that when you delete larger portions, especially of loops, extracellular loop number 2 and number 3, to some extent also number 1, you get this massive reduction in the ability of the bacterium to pull down vitronectin. And so based on these studies, we developed this molecular mechanistic model in which we propose that ale interacts with vitronectin to both promote the breakdown of blood clots into fibrinolysis and enhance the dissemination of the bacterium through blood, to promote cell adhesion through the interaction of vitronectin with integrins as cell membranes, and to disrupt the innate immune system through its interactions with this membrane attack complex channel. And so these are the activities of the proteins that we're trying to chase down. Now, one of the things that we've done, and this is the work of Kyung-Soo Shin, a postdoc in the lab, was to first of all determine the structure of the major C-terminal domain of vitronectin. It's a very pretty structure. You can see this 4-bladed beta propeller structure with calcium ions bound on the inside. Based on this structure, we were then able to repeat or pull down experiments with bacteria that express ale and show that the interaction of ale with vitronectin is in large part associated with this C-terminal domain, with this beta propeller. The antibodies show that, or they indicate that the activities are really localized to these loops of the vitronectin structure that form the edge of the beta barrel domain. Now, this is a crystal structure, it's not NMR. Apologies for that, but it's been very useful, and we have now NMR spectra of that structure that allow us to map the interaction. Okay, so back to the bacterial membrane. As I said, we really want to be here, but on our path towards a native cell environment, we focused on trying to reconstitute a bacterial outer membrane for NMR studies. So the bacterial outer membrane is highly asymmetric. And what I mean by that is that it's not a symmetric bilayer, where the two leaflets, inside and outside, have identical lipid composition. It turns out that the inner leaflet is made up of phospholipid, and that's shown right here. And the outer leaflet is made up of lipopolysaccharide. And the structure of a lipopolysaccharide is shown right here. It's basically composed of a lipid A component, which typically has 6 to 4, in the case of Yersinia pestis, 4 acyl chains, connected to an inner core, and then in some cases an outer core of sugar molecules. Now, in the case of Yersinia pestis, the LPS is restricted to an inner core, so it's a shorter version of the LPS that enables the loops of AIL to extrude to the outside of the phospholipid bilayer. So here it is, the structure of Yersinia pestis LPS. Now, the reason it's interesting to look at this is because Yersinia pestis has evolved both AIL and LPS structure in tandem. Okay, so the expression of AIL and LPS are highly synchronized, meaning that at 37 degrees, which is the human infection temperature, you have this massive production of AIL so that it becomes about, it composes about 30% of the outer membrane proteome. And also you have this downregulation of these LPS acylation genes, which basically make sure that the form of LPS is this tetraacylated form of the molecule. Okay, so at 37 degrees you get this structure. And so we asked whether LPS has an effect on AIL, and similarly, does AIL have an effect of LPS? You know, what does that membrane look like? And so I want to highlight here the work of Chandan Singh, who was a postdoctoral associate in the laboratory at the time and is now professor at, in the Department of Biochemistry at Banaras Hindu University. And so Chandan had the, so he embarked on this project to make first nanodiscs and also then liposomes where the membrane mimicked an outer membrane, that is, it contained LPS. And he worked with Yersinia pestis LPS, provided to us by our collaborators, Andrei Anisimov. And so Chandan had the bright idea to recognize that one LPS molecule has a surface area that's about 3 times as large as that of a phospholipid. And so he prepared these nanodiscs by basically replacing 3 phospholipid molecules with one Yersinia pestis LPS molecule. And what he ended up was these beautiful spectra. These are solution NMR spectra, proton, nitrogen, HSQCs of N15 labeled AIL in nanodiscs that do not contain an LPS in black, compared to nanodiscs that contain actual Yersinia pestis LPS. So you can appreciate that the quality is quite nice, and there was a lot of work put into this to make sure that the preparations are highly homogeneous using size exclusion chromatography. What's very nice is that by comparing these two spectra, Chandan could identify specific chemical shift perturbations that we could then map onto the protein. And so the map of the perturbations is shown here. It's mapped onto the structure in the right. And you can see that the highest perturbations shown in red and highlighted as spheres really map to, first of all, the extracellular loops as expected, but also these specific regions on the sides of the beta barrels. And interestingly, also to the inner leaflet, to some extent to the inner leaflet, to the intracellular regions of the protein. These were aided by molecular dynamics simulations, which we conducted in collaboration with Wonpil Lim and Lehigh University, and they were really useful for helping us to understand the mutual effects of LPS with on AIL. And so what we discovered based on the combination of these NMR experiments and MD simulations was that there are two patches of predominantly basic residues on either side of the Ale beta barrel called patch 1 and patch 2, which are really responsible for making these very specific contacts with the phosphate group and the sugar hydroxyls of the LPS right here. And so we really identified these as LPS recognition motifs on the surface of Ale. And indeed, when we went and mutated these various sites together with Greg Plano, we found that the interactions of Ale with LPS mediated by these cluster 1 and cluster 2 residues are really important for preventing things like cell leakage, shown up top, for promoting polymixing resistance, or resistance to antibiotic that's dependent on a disruption of the outer membrane, and for promoting serum resistance. So these residues in Ale are critical for establishing interactions with LPS and for ensuring that the virulence phenotypes of the protein are preserved. When we further look at the effects of this interaction, we compare Yersinia pestis outer membranes with Ale versus completely symmetric lipid bilayers and Ale. You can see visually that the extracellular loops of the proteins acquire extensive conformational order in the presence of LPS, whereas they are highly disrupted here. And this is highlighted in this chart at the bottom, showing that the root mean square fluctuations, the RMSF, in the presence of the, sorry, these are inverted actually, in the presence of the lipid bilayer are highly, so the root mean square fluctuations are higher in a symmetric lipid bilayer, shown in black, compared to Yersinia pestis outer membrane that contains LPS. So LPS has an ordering effect. And indeed, we did show this using solid-state NMR spectroscopy, and this was the work of Samit Dutta, who was also a postdoc in the lab back in 2017. Actually, before we had done these experiments, we had evidence that the addition of LPS to, in this case, phospholipid bilayers would really lead to this dramatic increase in conformational order that enhances the cross-polarization efficiency of our spectroscopy. So the data really point to this cooperative functionality of the Ale and LPS assembly, where the Ale in the presence of LPS establishes these interactions that are important for displaying the loops of Ale at the bacterial cell surface. And these, of course, are important for all the things that I mentioned, conferring serum resistance, maintaining membrane integrity, and supporting antibiotic resistance. Whereas the disruption of these interactions through mutations then leads to these floppy loops and to this expanded membrane. I don't have time to go into the effects on the membrane, but these are also significant, that basically disrupt all of these critically important functions for the pathogenicity. Okay, so where we really want to be is in a cell membrane. The question is, can we do NMR in native membranes? And you know, the new area of in situ NMR is advancing and very exciting. And the reason it's important, of course, is that we want to study these interactions in the native environment. And so the recent work of James Kent, I will show you, is really showing that yes, the one can do this for this specific system of veil in the outer membrane. So James has been developing protocols now for the native expression of veil in the bacterium. So all of the previous work that I showed you was purified protein that was expressed as inclusion bodies and then purified and refolded into a beta barrel. What James is doing is what James has done has been to develop a method where the protein sequence is expressed and translated into the outer membrane by utilizing the BAM complex directly. So the protocol basically involves expressing the protein in E. coli cells and then lysing the cells to isolate the entire E. coli cell envelope. Okay, and so you can see that if you look at gels of the cell envelope, you end up with these basically single band preparations of AIL. You can also isolate the outer membrane and have even cleaner preparations, although that involves detergent, and so for the moment we're trying to stay away from that. But the notable thing here is to see that the gels really show that the protein is folded, correctly folded in the outer membrane. That's a property of outer membrane proteins, that you can really distinguish folded from unfolded based on denaturing gels. So the SDS page really shows us that we have homogeneous preparations of the protein folded in the native bacterial membranes. And so what James has done is to take the entire cell envelope, that's right here, CE, which includes the inner membrane, the periplasmic layer, the peptidoglycan layer, PG, as well as the outer membrane. So you're taking this whole fraction, basically the shell that encapsulates the bacterium, shown right here as a cartoon, and put that into the magic angle spinning rotor for NMR experiments. And here are the data. We just submitted this to bioRxives, and the paper is under review. You can see that when we compare now the cell envelope spectra shown in black with the spectra that we had obtained previously from reconstituted ALN liposomes, you see these beautiful correspondences of the resonances, again showing that the overall conformation of the protein is similar in reconstituted preparations to actual bacterial cell envelope preparations. Similarly, this is also borne out in the proton nitrogen CP HSQC spectra from ALN liposomes and ALN cell envelopes. The notable thing here is that the AL liposome spectrum was obtained from a sample that was highly deuterated and hence the nice result lines, whereas the cell envelope spectrum was not deuterated. AL is not deuterated. So given that, this gives a lot of hope that much more could be done with this. Now, despite the fact that the spectra are highly similar, we can also detect specific differences that map to these red regions of the protein. Again, it's not just the extracellular loops, but the data really suggests that it's the entire membrane environment that has subtle effects on the entire beta barrel, that the beta barrel really senses the difference in membrane environment. So finally, to circle back to vitronectin and the interaction with human, the human host, what we really wanted to see whether we had whether we had generated a sample that was functional. And so what James did was he took his E. coli bacteria, in this case, expressing AL, I'm looking at the top figure, and what he did was to plate the bacteria on either heat inactivated human serum, HIS, here, AL plus and AL minus, or in normal human serum with all the factors, including vitronectin, that can be active in killing bacteria. And so what you see that in the presence of AL, the bacteria can survive, but deletion of AL prevents the bacteria from survival. So it's really confirming that the expression of AL in E. coli even is able to confer the serum resistance phenotype even to E. coli. Moreover, James showed that the bacteria that do not express AL do not form a biofilm, whereas AL plus bacteria have this specific pellicle formation phenotype where the bacteria can form a film, a ring basically, around your test tube. And finally, he was able to show that AL plus E. coli is able to pull down vitronectin. And here is vitronectin right there. Both vitronectin and the domain of vitronectin that we generated for structural studies. And so we have bacteria that now begin to recapitulate the pathogen, the virulence phenotypes of Yersinia pestis. And so what he did with that was to take the cell envelope preparations that he generated for NMR, incubate them with normal human serum, and then put the whole mix inside the magic uncle spinning rotor. And you can see now when we compare black spectra without human serum to blue spectra in the presence of serum, you can begin to detect these chemical shift perturbations that are very interesting because they really point to interactions of potentially direct interactions of the protein with vitronectin or other serum components. We don't know what they are at this stage. We have to figure that out. And also potentially, again, perturbations due to perhaps some interaction of serum components with the outer membrane. This is early stage work, so we obviously have a lot to do to characterize the interaction molecularly. So with that, I hope that I gave you an overview of the work that we're doing in the laboratory, of our mission really to try and understand these things in real samples and make and have an impact on potentially therapy development down the line. Multiple people worked with us on these projects and they're listed here. Again, I thank you very much for the honor of presenting to this meeting, and I'll take questions.
Okay, thank you, Dr. Francesca. Sorry for the initial technical glitch. Now, talk is open for discussions. Please raise your hands if you have any questions. Okay, Ashutosh. Keith, unmute Ashutosh.
Jeet, can you unmute Ashutosh?
So Ashutosh, you can unmute yourself and ask questions.
Yeah. Ashutosh.
Do you see him raising the hand? I don't see him.
Okay. Next question, Grace.
Thank you, Suri.
Yeah.
I was wondering about vitronectin. Very nice talk. How big is that? You said you mentioned that only the C terminus is interacting. Is it the only domain that has a role in the function of AIL, or the other domains are also have been shown to interact with this function of AIL?
So thank you for the question. That's a really important question. So vitronectin was discovered in 1967, I believe, and there has been a lot of work done on it, but it's been a very difficult protein to work with. The protein is 460 amino acids long, but it shows up on gels as a 75 kD band because of three glycosylation sites, glycosylation at 3 sites. The C terminus that we determined the structure for spans residues, like the last 300 residues or so, and we had to delete 2 loops that are predicted to be conformationally disordered. So we're now in the process of putting those back in. We're also in the process of trying to produce the full-length protein using baculovirus expression. So we don't know whether the C terminus is the only region that interacts with vitronectin. It's likely to be so. And we also think that possibly at least one of the loops that we deleted may be important for the interaction. So I think that that's an expanding story. And it is the one that's gotten us really excited about the biochemistry of vitronectin. You know, there's a whole other story, macular degeneration, which I think is fascinating. And also in the formation of deposits like the ones found in Alzheimer's disease that are really, really interesting. So I think that getting at the full structure will be important to follow up on.
Okay. Could you also comment on where the antibiotic is binding on AIL protein?
It's so the antibiotic, or
yes.
So the antibiotic is not binding to AIL, but it's disrupting, it's interacting with the outer membrane of the bacteria. And the interaction of AIL with lipopolysaccharide is important for conferring the strength to the outer membrane that prevents the antibiotic from disrupting that outer membrane.
Thank you.
Okay, let us thank Dr. Francesca. We are running late today, slightly. Thank you, Dr. Francesca, for your excellent talk.
Thank you.
We'll not take any more. We'll not take any more questions. We'll go to the next talk now, please.
Francesca, if you don't mind, if you can quickly do one thing, that somehow you became the host of the meeting. So do you see NMRS India there, and you can click on that and make the host? If you
where should I click?
If you click on NMRS India, that thing, if you see, and if you give a right click on the NMRS India profile, can you see on the gallery view?
No.
Yeah.
No, it's okay. I mean,
so in participants, you should see.
Yes.
Yeah, I see it. Make host. Okay.
You make host.
Somehow this you became host.
Okay.
If you make that host, everything will be all right now.
Okay.
Okay. Yeah. Fine. In the meantime, we'll go to the next talk. And next talk
Prateep,
is by.
Yeah, Prateep, you are the co-host. If you although your slides
Yes.
there, but
I am.
Yeah.
Okay. Next talk is by Dr. Prateep
Oh,
Bhattacharya.
that's great. Yep.
Okay. Prateep is from. He did his PhD from California Institute of Technology. He did his postdoc in the year 2005 and 2006 with James G. Boswell Fellowship. He worked on MR physics and Huntington Medical Research Institute, Caltech. His primary appointment was as an associate professor, Department of Cancer Systems Imaging, Division of Diagnostic Imaging, the University of Texas. And he had a number of other joint and adjunct positions. He's a graduate faculty, Division of Graduate School of Biomedical Sciences, the University of Texas Health Center, Associate Professor, Department of Imaging Physics, Division of Diagnostic Imaging, the University of Texas. And his main focus of research is in the Department of Cancer Systems Imaging is the development of real-time metabolic and imaging applications by hyperpolarization. Now, over to you, Dr. Prateep. And you can please take 25 minutes and leave 5 minutes for the discussion.
Yeah, absolutely. Thank you. Thank you for the very kind invitation. And before I actually start, let me see if you guys can actually see my screen.
Yes, we can.
Are you all seeing my screen?
Yeah.
Okay, awesome. Awesome. Thank you for the invitation. It's a great honor. And before I actually give my talk, I just want to take 30 seconds to pay my essentially honor and respect for, let me see if I can actually go to the next slide. Just a second. Yeah, to Dr. Hanudata Atreya. The short background is I it is quite unfortunate that I didn't have the chance to see him. One of his postdoctoral or graduate student actually joined my lab, Dr. Shivanan Pudakalakati, and he is an outstanding student. And I tried to meet him during the NMRS meeting in Delhi last year, and he was not there at the time. And he essentially emailed me that for giving this talk for this seminar session. And I was really hoping to catch up with him, but very unfortunately, he passed away at a very young age, and he's a brilliant scientist. So it's really unfortunate that I couldn't have a chance to meet him, at least virtually. So essentially, he's the one who invited me to this seminar. So I'm really regretting that I lost this chance. But with that, I'm going to give a talk on the metabolic imaging, what my lab does. To give you a short background, again, let me see if I can scroll the screen. Yeah. So I just want to show you why we do what we do. Essentially, my lab does NMR and magnet MRI, but MRI by itself has been around for last 30 years. And despite its popularity, there's one fundamental problem in MRI, which is it lacks the sensitivity. So this slide essentially shows the penetration depth. So just to give you an idea, human being, we are like a large animals. So like people are used to confocal microscopy where the penetration depth is really great and it's a very high resolution. But with MRI, we can go deep. For deep tissue imaging, it's a great opportunity. It can go deeper. But as it goes deeper, sensitivity is very poor. So right now, conventional MRI, we can see resolution in the 1 millimeter range, while to give you an idea, confocal microscopy is 1 micrometer range. So there is a huge chasm of sensitivity, which can be improved. So this is an opportunity for us. We can increase the signal of MRI in some way, then you can actually probe biomedical processes in real time and with great sensitivity. And with that came the idea of hyperpolarization. You can see how I can slide the screen. Just a second. Yeah, so NMR by itself is not sensitive enough because it only captures few parts per million of the spins. And it's not fast enough because if you ever volunteered as an MRI scanner, you'll see it takes almost half an hour to scan it. And it's not cheap enough. It's like a 1 million per Tesla. So just to give you an idea, there's like almost 100,000 1.5 Tesla MRI machine all over the world. I'm talking about human scale and MRI. And only like 3 or 4 9.4T. And the price is like quite an order of difference. 9.4T is like a $9 million each. And the sensitivity is higher. It's like a couple fold, but it has other problems with noises and stuff. So it's a very expensive way of doing it. The next slide, please. So essentially what my lab wants to do, we want to increase the both sensitivity and specificity of MRI. And we do that by a variety of technique, but one of the things I want to focus on today, because given the lifetime limitation, is essentially metabolic imaging using this technique called hyperpolarization. And I want to show you one specific example in pancreatic cancer because it is one of the most deadliest asymptomatic cancer known to mankind. So next slide, please. This is an interesting way to explain hyperpolarization. I think most people are familiar with it. So in conventional NMR or MRI, the spins are thermal equilibrium in Boltzmann distribution. So the spin difference is like few parts per million. So once we, can you press the next button? So it'll probably show it. So essentially spins cancel each other and we get this sensitivity. Press the next button again. We should be able to see a signal, which is fine, but in NMR spectroscopy, you can average the signal over a period of time and you can get a reproducible high signal. But in MRI, unfortunately, the patient tolerance is a big problem. And you also cannot do the technique like spinning. You cannot spin a patient, that's not possible. So we get a very low signal. So in hyperpolarization, we artificially create these spin differences. So next button, please. So again, when we cancel each other, so we get a signal, again, press, this is a cartoon animation. Sorry, I don't have access here. So you get a signal which is at least 10,000-fold signal enhancement. And we call this process hyperpolarization. So with this, we can now interrogate biomedical processes which cannot be done before in real time and in live intact animal system and hopefully in human. So next slide, please. So essentially we are increasing the size of a quarter to an Empire State Building. So I was almost tempted to do that like a small Indian paisa to like to the sign of a Qutub Minar. So it will be like a huge signal enhancement. So we call it just a way of showing that. The next slide, please. Since this is more NMR crowd, I want to just show you the periodic table. So the different nucleus we can potentially hyperpolarizable. Many of them you can see by marked, which is like a fast relaxation. So for interrogating biomedical processes, we need nucleus which are long-lived signal, long enough to capture a real-time metabolic signal. So for that reason, so hydrogen would be a really great choice, or fluorine could be a great choice, or phosphorus, but they relaxes really fast. And so you can't quite use them. So fortunately, carbon is in the place where it is not great, but enough relaxation time of a couple of minutes to allow us to interrogate biomedical processes. So on a separate, this is a complete different talk. Part of my lab also works on hyperpolarizing silicon particles for targeted nanoimaging. And in silicon, the signal lasts over an hour. So there you can store the signal almost like a spin battery concept. But that's beyond the scope of the talk. But I'm going to show you, there are many NMR and chemists here, so they can think of new nucleus for hyperpolarization imaging. So for this talk, we'll be entirely focused on carbon imaging. Next slide, please. So there are multiple techniques of hyperpolarization, parahydrogen-based, which is essentially discovered by my former mentor, Dan Whitekamp at Caltech. And part of my lab used to work on this. But the problem is, for biomedical system, there is a pathway for human transition is really poor, because every time we are actually reacting with hydrogen with an unsaturated bond to create a new molecule. So our federal FDA will not allow us to take it to the clinic because there's no guarantee that we can make a stable compound at every point. So there are other techniques, the hyperoptical imaging with xenon hyperpolarization. And there's a new technique emerging called brute force. In fact, there's a company in Boston area, a startup called Millikelvin Tech. They're trying to make nucleus polarized at using very low temperature and high magnetic field. They call it brute force hyperpolarization. But the one which is very easy, which is most common right now, is DNP, dynamic nuclear polarization, where you can polarize a solid state at a high magnetic field and low temperature, and then quickly add a liquid, in this case hot water, to essentially dissolve it and inject it in an animal or a patient. So all the talk I'm going to show you is all about DNP hyperpolarization. Next slide, please. So just keep in mind, I want to show you for sake of completion, so that we need to, for biomedical imaging, we need to really be cognizant of the fact that a cardiac output of our human system is 6 liters per minute, while the injection rate in usually 10 ml per second. So as we inject it, this is shows the time it takes different to reach different organs. So keep clicking on this slide, please. So you'll see it reaches heart at like 3 to 4 seconds, coronary artery in 15 to 20 seconds, brain in like 23 seconds. So we need a T1, the relaxation time should be at least 20 seconds. So keep in mind that's really important for our carbon molecules to show that. So the next slide. So with that hyperpolarization, we now have the ability to do experiment really fast and cheaper, and do longitudinal study and do real-time metabolic imaging. So I think it's almost self-evident, but I want to show you this just to for a point of completion. Next slide, please. So the reason we want to image metabolism is it's pretty well known that cancer cells have sweet tooth. Essentially, this is a well-known Warburg effect. And the next slide. So in a normal cells, I mean, I'm assuming there's some biochemist here, normal cells, when glucose go into the metabolic cycle, it is metabolized to pyruvic acid and then to TCA cycle to carbon dioxide and water. And that's a well-known glycolytic and TCA cycle. But only when you're working out or doing a running or muscle building or weightlifting, your muscle gets sore, and this is under oxygenation, and you produce lactic acid, which causes the soreness in the muscle. And this is well known. However, in cancer cells, irrespective of the oxygenation status, whether you're working out or not, cancer cells take the pyruvic acid and produces lactic acid. So this was actually well-known phenomena observed by Warburg, who escaped the concentration camp in Germany. in 1924, and right now, in last 10 to 20 years, there's a revival of this metabolism led by Van Hander Hayden at MIT. There's a lot of, he's a cancer biologist. There's a lot of work, and in fact, reinterpretation of metabolic imaging going on all over the world. But what I want to this is beyond the scope, but keep in mind, this is what we observe. In normal cell, it goes through the TCA cycle. We do not see that much lactic acid, but in cancer cells, it goes through the lactic acid pathway, and this is Warburg effect. Next slide, please. So what we do in this experiment is we essentially hyperpolarize the carbon-13 of C1 pyruvic acid. So once you do this, and since NMR is a spin property and it can survive chemical reaction, so we can actually image, and by both spectroscopy as well as chemical shift imaging in MRI, the downstream products of the molecular we're hyperpolarizing. So in this case, pyruvic acid goes to lactic acid, and you can able to see, because lactic acid is preserved in the C1, and you can able to see the lactic acid signal there. And it can also go to alanine, to a different pathway, and it from carbon dioxide and water as well. So keep in mind, so you can able to image them because the carbon-13 hyperpolarized signal is preserved in the downstream product. Next, click. So this is what we expected to see. I want to give you an example of that. So this is a real-time spectra, serial spectra take after 1 second each. So essentially you are seeing real-time evolution of the metabolism. So this is hyperpolarized pyruvic acid injected in a tail of a rat, and you're looking at the liver of a normal rat. What we see, this is the huge signal of pyruvic acid. Then this is a normal rat, so all you can see is the lactic acid and small amount of alanine forming. And each of the transient are taken after 1 second. So you can actually see the real-time evolution of the metabolism as it is unfolding in a real, live, intact animal. So it's not an indirect visualization, it's a direct visualization of metabolism as it is unfolding. Next slide. So now so this has been discovered by Goldman. So in fact, the original discovery of the DNP process by Overhauser several years ago in the 50s. But there's a revival in biomedical application by Claes Goldman at Amersham in early 2000. And then it is taken up by different labs, this DNP hyperpolarization. So my lab essentially uses this for interrogating pancreatic cancer. And I'm going to come to it in little later, why pancreatic cancer. But before I do, I want to show you, we essentially follow 2 different modalities here. One is we inject hyperpolarization in a pancreatic cancer mouse, and we see exactly the same thing, like we follow the metabolism as it is unfolding in the pancreas in the mouse, in a live mouse. Then we sag the mouse, and this is a kind of a gross image of this animal, and this is the pancreatic tumor. Then we take the tumor out, flash freeze it, and then we do a complete NMR-based metabolomics. So essentially take the flash frozen tissue and then extract the metabolites, and at a high resolution NMR spectrometer, we collect the NMR spectra. So one is a dynamic, this is the real-time live intact imaging inside the pancreas of a live mouse. The other one is a static image of the tumor tissue. So next slide, please. So we compare it like the static image is like a cars in a parking lot. We are counting the number of cars. So it's a static image of the ex vivo tissue, which is a global metabolomics. But the hyperpolarization gives you the dynamic state of the imaging of the metabolism as it is unfolding. This is like a cars flying in the freeway. So there are 2 different images as you can get simultaneously in an animal. Next slide, please. So for this talk, we're going to focus on 3 different areas, and I'm going to give you some flavor already published and some on the work we're working on. And 3 areas, one is the early detection of pancreatic cancer. Next one is how you can interrogate the aggressiveness of pancreatic cancer. And the third one is that how can you image the early efficacy of therapy in pancreatic cancer. So I will start with the first one, the next slide, and the next slide. So pancreatic cancer is one of the deadliest cancer. The reason I got interested in it, because pancreatic cancer is completely asymptomatic. So those whose family member have gone through pancreatic cancer can attest to the fact that there's no signal, no indication, and suddenly they were bloating and they have jaundice, and they found there's a full-blown pancreatic cancer. And unfortunately, even in stage 1, I'm going to emphasize that even in stage 1 pancreatic cancer, the chances of survival 5 years is less than 10%. It is, in fact, Many scientists and oncologists and cancer biologists think that stage 1 pancreatic cancer is already a full-blown metastatic pancreatic cancer, also already micrometastasis going on, and there's nothing we can do. However, how the pancreatic cancer progresses is really interesting. So there's a top of the slide I want to pay, even though many of you are not biologists, I want to focus on this top of the slide. Is really interesting in that way. So in normal pancreas, the cells are like this. It goes through some inflammatory cycle. Yes, thank you. The slide before. Yes, so it goes through a normal inflammatory cycle. We call it PANIN1. And this is completely reversible. There is no way of detecting it at any point by MRI or any other way. And it goes through something called PANIN1B and then PANIN2. While it happens, the mitochondria and becomes larger. And it goes to PANIN3. So the hypothesis right now, or the biologists, what they think, or PANIN1, PANIN2, PANIN3 are completely irreversible. They're moving back and forth. But somewhere in the process, it moves to PANIN1-3, and then it moves to the early stage pancreatic cancer, which is irreversible at this point. So which is invasive pancreatic adenocarcinoma. And at this point, there's no way you can detect them. Only discover is incidental finding when you're operating or hysterectomy. That's one of our Supreme Court justice just died last year in the United States, Ruth Ginsburg. In fact, her pancreatic cancer was discovered as an incidental finding in 1990s, just when she was doing her hysterectomy, and one of the surgeons saw this PANIN, and they removed it, and that's why she survived that long. And she died of metastatic pancreatic cancer last week. Is absolutely tragic. But the fact that she lived that long because her surgeon was able to see this PANIN while she was undergoing hysterectomy. If you can, so essentially, if you can image these stages by MRI, then you can actually save pancreatic cancer patient. So this is enough of a background. But what I want to tell you, one of my colleagues at MD Anderson, I'm going to talk about her a little bit. Her name is Florencia McAllister. She's an absolutely motivated pancreatic cancer oncologist at MD Anderson. And she's a collaborator. Part of that, her mom died of pancreatic cancer at a very early age. And she has the genetic markers to make her as a high risk. So she's a very motivated pancreatic cancer oncologist. So her lab has developed this genetic engineered mouse model of pancreatic cancer early stages. So what I mean is that her mouse animal model, these are animal mice, which recapitulates this early stage of PANIN1, PANIN2, PANIN3, and full-blown progression to pancreatic cancer. So when she has that, I thought it's a great opportunity that you can use this metabolic imaging to, can we find any metabolic marker? Can we image these mice at an early stage? So next slide, please. So what we did, so this is a busy slide, I want to keep your attention. So the top one is a normal mouse. 10 weeks is a PANIN1 early stage pancreatic cancer, not even early stage, it's an early stage pancreatic inflammation. 20 weeks is later stage, still PANIN, and 30 weeks when it's a full-blown or just early stage pancreatic cancer. So from 10 weeks and 20 weeks, they are not cancer, but they're early stage inflammation. So what we did, and the middle slide shows the histology. I mean, you don't have to understand, but for the cancer biologists, we can actually see that really, what is the proof that they're actually become towards cancer. So you can see that there. So what we did, we inject hyperpolarized pyruvic acid to the tail vein of these mice at different stages. And what is amazing, what we saw, we can able to detect hyperpolarized alanine, hyperpolarized lactate. So in the early stages of the normal pancreas, we see a lot of alanine and no lactic acid. But as we can image them in real time. And then as it is progression towards the pancreatic cancer, the alanine is going down and lactic acid is going up. And we can able to see this in real time. And in fact, as a proof of concept, we even took the tumor tissue, sacked the mice at this early stage at different time points, and look at the conventional NMR, proton NMR spectra taken from the tissue sample. And this is what you see. The alanine is very high. You can see the proton spectrum. And this is, and hyperpolarized is all carbon-13. So you can actually see alanine is decreasing as it becomes more and more towards the pancreatic cancer. And lactic acid is, alanine is decreasing and lactic acid is increasing. Next slide, please. So this is how the imaging is done. So we know the general region of the pancreas. So we put a slab. We can do that virtually. And then we inject the hyperpolarized pyruvic acid. And this is the hyperpolarized spectra. This is a relative intensity. top one is the pyruvic acid, the bottom one is the lactic acid, and the middle one is alanine. I know this is kind of a busy slide. So this is the control, means normal pancreas. But look, as we move towards pancreatic cancer, pyruvic acid, the alanine is decreasing and lactic acid is rising. And in fact, we also measured the alanine to pyruvic acid conversion and lactic to pyruvic acid conversion. And we can see that the rate constant is also different. So what we're now proposing, the next slide, please. So alanine to lactate and pyruvate to lactate ratio and the rate constant. So pyruvic acid has two different fate. It can go to alanine, it can go to lactic acid. So there is a competition as it becomes more towards the pancreatic cancer, more pyruvic acid is shuttled towards the lactic acid and less towards the alanine. And this can be a great biomarker. So with Florenzia, next slide, please. So this is Dr. Florenzia McAllister. She's the first one in the world to create something called high-risk pancreatic cancer clinic at MD Anderson. What that means is the patient who have a family history of pancreatic cancer or known genetic mutation, they come here, they do not have cancer, and they come here to MD Anderson Cancer Center, which is the biggest cancer center in the world, to get scanned. And we are now trying to do a clinical trial for detecting that can we see the same metabolic changes in real time in this patient. It's a very expensive way of doing experiment. But if we are successful, then we can actually make the new marker by which you can detect metabolic changes for early detection of pancreatic cancer. So this clinical trial we're about to start, and we are now going through the FDA approval process, and Florenzia is leading the clinic of that. So we do not have the data yet. Hopefully at some point soon, we will be able to show you human data. So with that, the next slide, please. So what I've shown in this slide, that lactate to pyruvate and alanine to lactate ratios and the rate constant can be a biomarker for early detection of pancreatic cancer. Next slide, please. So now comes the assessment of aggressiveness of pancreatic cancer. Next slide. So this is, again, MD Anderson, we are very fortunate that there are a lot of patients come here at MD Anderson with pancreatic cancer to do their surgery. So these are already patients have pancreatic cancer. So one of my colleague, Jason Fleming, who is now at the head of pancreatic oncology at Moffitt in Florida. So he developed this mouse model where he took the tissue from the patient. He's a surgeon. He did the surgery on the pancreatic cancer patient. And then he grew this same tumor, which is taken from the patient, on mice. So he developed this pancreatic cancer patient-derived mouse model. Essentially, these mice recapitulate the same pancreatic cancer aggressiveness and other profiles, which are already there in the patient. So next slide, please. So we selected 4 types of mice, each n equals 5. One, we call it MDA-PTX148. This is a patient which is super aggressive pancreatic cancer. And you can actually see, even in mice, look at this this red line. Even in mice, the tumor grows and the mice dies in months. While PTX142, which is a much slow growing pancreatic cancer in this patient, which actually goes much longer time. So here we selected this 4 group of mice, and we first did an NMR metabolomics. So we sat the mice, took the tumor, did NMR metabolomics at a given time. And this is fascinating. You can see in the most aggressive pancreatic cancer mice, we see the highest amount of lactic acid, which makes sense because the more aggressive it is, it has the more Warburg-ish metabolism is going in that direction. So you see high concentration of lactic acid. But remember, this NMR spectroscopy by the ex vivo tissue samples essentially measures the total lactic acid pool in the tumor. So this is not the flux, but the pool. As I said, cars in the parking lot. We are just counting the number of cars in the parking lot. So the next slide, please. So this is the hyperpolarized metabolic imaging. So I want to show you, this is actually so fascinating and dramatic. So this is the mouse with a tumor in it. So we put a Fox cell in the tumor and we inject hyperpolarized pyruvic acid. So what we're essentially doing, we are measuring the real-time rise of lactic acid in the tumor of these 4 different patient-derived animal models. Now I want to bring your attention to PTX148, which I said it is the most aggressive pancreatic cancer mice. So in the patient died within months of having the cancer, and the mice has the same result. So look at that. So we're injecting pyruvic acid, and pyruvic acid is converted to lactic acid. Just imagine there's a huge metabolic machine sitting inside the system of the mice. The pyruvic acid never even have the time to grow. So each of the segment is seconds. So within seconds, pyruvic acid completely consumed and is formed to lactic acid. We are able to measure that in real time with hyperpolarization and within seconds. So the moment you inject pyruvic acid, is gone. It is not even time to build up and it is converted to lactic acid. Now take the least aggressive mouse model. So when we inject pyruvic acid, it has enough time to grow slowly, and then we see the rise of lactic acid. Next slide, please. So when you do the math, you can see in the most aggressive mouse, the completely solid dots represent the rise of pyruvic acid, red, and the hollow dots are for lactic acid. You can see pyruvic acid comes in and immediately there's a huge rise of lactic acid. And this is within seconds. The time axes are in seconds. And the least aggressive mice, it is very slow growing. Next slide, please. So this is the most interesting take-home message. So what one of my postdoc, Prasanta Datta, he did the work. So he plotted the lactic acid pool we measured from the NMR spectroscopy of the ex vivo sample with the hyperpolarized lactate to pyruvate ratio. And these are n equals 5 each family of categories of 4 mice. And what we see is a perfect straight line. What that means is essentially the pyruvate pool essentially recapitulating the total lactic acid flux. So essentially, we don't even need to do biopsy now. If you have a patient coming in, pancreatic cancer patient, we just inject hyperpolarized pyruvic acid, just measure the lactic acid concentration, and we can actually put them on this curve and say how aggressive the tumor is. So this is almost, it may potentially obviate the need of biopsy, which is very painful in pancreatic cancer. So we have a direct way of measuring real-time aggressiveness of pancreatic cancer. And you can actually see, so it was a cover story in Journal of Proteome Research last year. So next slide. So this is just, we also measured the different protein which was expressed, and it makes those who are interested in biological interest. So we can see that LDHA, which is the enzyme which interrogates the process, highly elevated in the most aggressive mice. And HIF1 alpha, which is the measure of hypoxia, it is also elevated. And there's other biological factors which we can attribute to why the mechanistic reason why this changes. But this is for this audience, what I want to draw your attention, next slide, is that hyperpolarized lactate to pyruvate ratio can be biomarker for aggressiveness of pancreatic cancer. The next slide. So now we'll talk about, because we're short on time, on the therapeutic efficacy of pancreatic cancer. Next slide. So again, so those who ever got cancer, they go through chemotherapy or radiation, and they have to wait for months before they know actually if their treatment is progressing or is responding at all in the first place. And it's a huge problem for clinicians and oncologists. We call it something called therapeutic dilemma, because we don't know if it's working or not. If it doesn't working, it is a wastage of time. So we can do other things and give the patient more chances. So what the idea is that, can metabolic imaging, hyperpolarization, can it detect early efficacy of therapy? And I'm going to show you the answer is yes. So this again in pancreatic cancer, what I've shown you before, pyruvic acid goes to lactic acid, you can measure that. So what we did, we took a drug which blocks this LDHA pathway. And we treat this cohort of mice and we measured the hyperpolarization at each different time point. So I bring your attention to before treatment. So when you inject hyperpolarized pyruvic acid, this is a full-blown pancreatic cancer. You can see this huge signal of lactic acid rising from the tumor itself. And we treat them for 4 days. And then day 4 of the treatment, we do the hyperpolarized metabolic imaging again. And look here, the lactic acid has gone down. So essentially the tumor is working. And in day 4, lactic acid is almost gone. So all this is happening when the anatomical tumor volume is not changing. So what is interesting is even the anatomical volume, the tumor is not shrinking, the underlying metabolic processes is already changing. So you can be able to see that in real time. And this is in a case where mouse is not responding to the tumor. Next slide, please. And this is a case when the mouse is not responding to the tumor. So here also the day 4, you see lactic acid is very high at day 0. And in day 4, you see there's no reduction in lactic acid. And on day 6, lactic acid is the most prominent signal. So here the treatment has failed. So we can able to distinguish between mouse which is responding and which is not responding. And we can do that within a matter of days. So if we do it on patient, we do not have to wait for months to get an answer. And this is why metabolic imaging can be very efficacious for early response to therapy. Next slide, please. And I want to give another example from in-home. This is unpublished data. And because the drug is much more interesting, this is our MD Anderson has an in-house medicinal research department called IACS. And they have developed this drug called IACS 10759. And that itself is a whole other talk. But what I want to tell you, this drug essentially blocks the OXPHOS pathway in the metabolic pathway. So if the OXPHOS pathway is blocked, what we see is the ATP goes up and the lactic acid goes up. And the reason of this drug is that the idea is that in pancreatic cancer, when the first line of chemotherapy always fails, because some of the pancreatic cancer cells, which are most aggressive, they don't die at all. So the idea is that this particular drug will be the second line. It will kill those aggressive cells, and that will make pancreatic cancer more enable pancreatic cancer patient to respond to therapy when the next chemotherapy cycle begins. So we want to know if this drug is efficacious in essentially reducing the tumor burden. So next slide, please. So if the drug actually is working, we should expect to see the reverse effect, means lactic acid will go up. So this is exactly what we see. This is the top figure is the two different kinds of pancreatic cancer orthotopic mouse model. And this is before treatment, the NLAC is the ratio of lactate to pyruvic acid. So NLAC is a lot lower before treatment, and after 4 days after treatment, lactate acid essentially goes up. And this is highly statistically significant. So you can able to see that in real time. Next slide. So this is a very short time, I know, but I'm going to quickly touch this. So my colleague at MD Anderson, who recently won the Nobel Prize 2 years ago in medicine, James Allison, for his revolutionary discovery of immunotherapy. And next slide, please. So immunotherapy is very exciting because it helps quite a bit of patient, but a lot of patient doesn't work. So this is a slide from New England Journal. People have published that even though a lot of patients respond to immunotherapy, there is a subset of patients who do not respond to immunotherapy. And this is a big question among the clinician. Can we image early on before you put the patient on this very expensive drug, immunotherapy? Can we say which patient is responding, which is not, will respond to immunotherapy in real time? So next slide, please. So the question is, can we do image and in vivo metabolic imaging biomarker for resistance to immunotherapy? Next slide. So this is in collaboration with Dr. Allison's former postdoc, Mike Curon, and this work is done by Shivanand, who is the postdoc working with Dr. As a graduate student, he worked with Dr. Atreya. So there's a mouse model which progressively becomes immunotherapy resistant. And at this point, it is only melanoma, we can get that. So the first group is completely respond to immunotherapy, only a small fraction doesn't respond. The red one doesn't respond. So as we breed them in progressive breeding, by the 4th generation, they become completely resistant to immunotherapy. So it becomes a very exciting animal application to check this out. So give me a next slide, please. So we took the 1st generation and we call it responding, and the 4th generation, which is resistant. And then here the same thing, we inject hyperpolarized pyruvic acid, and we want to see the NLAC, the lactate to pyruvate ratio, is that a biomarker of immunotherapy resistance? And lo and behold, we saw the exact same thing. In immunotherapy responding mouse, the lactic acid is a lot higher, and immunotherapy resistance is a lot lower. Next slide. So, in fact, I take it back. In immunotherapy resistant, lactic acid is a lot higher. It makes sense that it is not working very well. And the one who respond, it is a lot lower. So we have published recently that immunotherapy research is just coming out in press. So again, hyperpolarized metabolic imaging can enable us to detect early efficacy of both chemotherapy as well as immunotherapy. Next slide. So that's the whole point I'm making. This next slide, please. And this is, I'm almost close to an end. And so I think Dr. Moresi gave a very exciting talk touching on some of the microbiome. And my colleague Florencia McAllister just published a very well and highly rated paper right now in Cell last year, where she has shown that in pancreatic cancer, microbiome plays a very important role whether the patient is responding to chemotherapy or not. So now we have also, there's a published paper by other labs that microbiome also changes the metabolism. So one of the projects which Shivanand is leading, and I do not want to share the data yet because we're still working on it. The question is, can the microbiome changes the metabolism, and does hyperpolarization with this huge signal enhancement can capture the changes in metabolism due to microbiome? So that is a very interesting project, but we are trying to recapitulate, or not recapitulate, to measure or image the metabolic changes in microbiome by the changes in drug, and does it alter the metabolism in real time? That we do not have any data yet. Hopefully in the next talk, I'll be able to share that with you. Next slide, please. So this technique is already in the clinic, which I told you. This is a slide from UCSF. They have shown that in prostate cancer, lactic acid is high, which you already know that, but this is in patient. Next slide. So at MD Anderson, we have the clinical polarizer, which was installed, and we're starting the first human trial in prostate cancer in September. And hopefully you can have the high-risk pancreatic cancer trial sometime early next year once you get the approval. Next slide, please. So in all, what I showed you, that this metabolic imaging with this hyperpolarization MR, which increases signal by huge, 10,000 to 100,000 fold, we can actually image different function of oncogene activity. So it is a really amazing modality of metabolic imaging in the future. Next slide. And this is a slide from Nature Review by a different lab. They show different drugs which are now coming up in the market which targets metabolism. So the idea is that we can able to see and use this drug to interrogate those processes. So the future of it is very exciting. The next slide. So again, this is the entire slide of metabolic cycle in human. And at this point, we can only touching just a really small section of it. The hope is that with hyperpolarized metabolic imaging as well as NMR spectroscopy, we can now expand that more, and we can touch this completely untapped encyclopedia of metabolism, which is completely untapped at this point. This is a great exciting opportunity for students and researchers who want to apply hyperpolarized NMR spectroscopy and imaging in this field. Next slide, please. So I call it this, it could be the discovery of the century. It depends on how far it goes. It's a funny slide. The next slide, please. So my hope is that sometime in near future, we'll be able to image that in real time. Next slide. And this is my lab. The most of the work was done by Prasanta Datta. Shivanand is doing the immunotherapy drug, as well as some of the microbiome work, some of my graduate students and postdocs. Next slide. And this is extremely collaborative research from multiple group from MD Anderson, Rice University, and multiple funding agency. And thank you very much for your attention. Next slide, please. That's it. Thank you so much. I'm happy to take any questions.
Okay. Thank you, Dr. Pradeep, for an excellent presentation. It was really enjoyable. So now the talk is open for the discussion. If you have any questions, please raise your hand.
I have a quick question.
Can I ask?
Chandra Kumar, please.
Yeah. You mentioned that one hopes to have assays under hyperpolarization conditions, but that would appear to assume a uniform hyperpolarization across different metabolites, for example. How realistic is that?
Yeah, great question. Actually, so right now we can only, so there are several labs that are working on hyperpolarizing different metabolites. So all the data I showed you today is only one molecule, which is pyruvic acid. So pyruvic acid, we are getting very good at polarizing at very high level, almost 60% hyperpolarization. So we can do that really well. So all the data I showed you is from only one molecule, which is hyperpolarized pyruvic acid. But you're exactly right. So as different labs are working on it, people are trying to polarize glycine, glutamine, and the challenge remains they're not uniformly hyperpolarized. And that is a problem. And I think as a community, we need to come together to find a standard for it. And that's why I didn't show you the data for, we have some work on that, but it is non-standard. So that's why I'm not comfortable in sharing that, but you're exactly right. And so pyruvic acid is now the gold standard for hyperpolarized imaging. So I want to show you the clinical application of that. But you're exactly right. At some point, we need to come to a standard, develop a standard for that. What is acceptable? Thank you.
Okay, question by Jeet. Jeet wanted to ask a question.
Yes, please.
Okay.
Jeet?
Can I go ahead, Jeet, if you?
No, I didn't. Actually, Vivek wanted.
Okay.
Oh,
So
Vivek, okay.
Hi, Pradeep. It was really very interesting talk, and it's always fun to hear your dynamic talk. Yeah. So I have quick two questions. So what is the concentration of the pyruvate that is being injected? Is it somewhere around the physiological concentration, or is it?
Yes, exactly. Well, yes and no. So that's a great question. In fact, there's a lot of debate on that because on the issue of the changing the metabolism. So for animal, we use like a very small, like 100 millimolar concentration. Then it undergoes tenfold dilution in the blood. And so in fact, even less than 100 millimolar. So once you inject it, if 100 millimolar become 10 millimolar, or 80 millimolar become 8 millimolar. So this is the effective concentration which you are measuring. So in human, there are some debate. In fact, we just published a paper in PNAS, which just came out last month on, this is not really a biological question, that what we found that in many cancer, the concentration could be a problem, but it also could be like how we are getting it. Means, remember, pyruvic acid is taken inside the cell by a transporter called MCT, monocarboxyl transporters, MCT.
Correct. Yeah.
So what we have found that many earlier paper on hyperpolarization. So in my graduate school days, I was actually my postdoc as an assistant professor, I was working on more the technical development on hyperpolarization. So I'm working on the biomedical system only recently. But several labs who have published papers on hyperpolarization, they're attributing the change of lactate level to some cancer function. But what we found that in the PNAS paper, that it is more a function of how much MCT is there in the system rather than a cancer function. So if the MCT is uniform, if the transporter which transports pyruvic acid to the cell, if it is uniform, then you are actually, it's a function of what the cancer is, aggressiveness or whatever that is. But if it's not uniform, you're actually not capturing anything else, but you're actually capturing the MCT concentration. So it's a bit of a controversial paper. So some people who are already invested in it may not agree with that. So I think your point is well taken, that the question is, if we increase the concentration a lot higher, does it essentially limited by the cancer function we're trying to image, or it is just a transporter function you are imaging?
But rather, I was trying to point out in the direction of the reaction, because we are not trying to propel the reaction in one direction by giving more reactant. And then like lactate synthesis is happening because now there's a lot of pyruvate which is present in the system now. So that's the kind of like idea that
that's a very pertinent question. And this is I think a lot of people in the work in this field are grappling. We try to address some of that in our paper which just came out, that MCTs, the transporters and the role in imaging. And in fact, we may end up seeing only measuring the transporter activity than cancer activity. That will become
okay. I'll just quickly move on to my second question and then I'll wrap it up.
Yes, please.
So
yes, please.
When you showed a slide where like in the early stage, you did not see the lactate formation, but when the cancer grew and progressed, you saw lactate production. So but in the initial stage, you saw alanine production, I guess, right?
Yes, exactly.
Like at that time, did you get only when the cancer was just in the initial stage, did you get only the cancer tissue or around the normal tissues were there, or the cancer progressed and you had pure cancerous tissue?
So let me, well, I'm not framing it right. It's entirely my limitation of framing it. So these are not cancer. So pannin 1, pannin 2, pannin 3, these are not cancer. These are, we call it inflammation. So only from pannin 3, and there's a lot of debate in this cancer biology, when pannin 3 progresses to early stage pancreatic cancer, there is a school of thought, they believe it becomes completely reversible, and there's another school who doesn't believe it. So essentially, but people agree that pannin 3 ultimately becomes early stage pancreatic cancer. So pannin are not cancer, they're just inflammation. And so there's nothing to image. So there is no tumor, we can image it. So what we know the area where the inflammation is, because it's a well-studied animal model. So we take the tissue around that area, so there's no tumor at all, and then do the processing and then measure the NMR spectroscopy. So there's no tumor at all. It is just the tissues from the pancreas, which we believe to have inflammation. But in the last stage, there are tumor. You can see nodules, even in proton NMR images, which are becoming more tumorous. So we can take that area and do NMR, then it's more on the tissue side. But in the early stages, there's no tumor at all. It is only tissues.
Right. So that could be the one reason that why do we see alanine rather than no lactate?
That is correct, but there's another question here. So other tissues, other non-pancreas do not have this process. So if you take a tissue, kidney, for example, or liver, you can see some alanine, but not this high concentration of alanine. So one of the other area we worked on is, so pyruvic acid can go to alanine, something called ALT, alanine transferase, the enzyme, and pyruvic acid goes to lactic acid to LDH, lactate dehydrogenase. So there may be some competition going on only in pancreas, doesn't have to be pancreatic cancer, between alanine and lactic acid. So probably measuring ALT and lactic acid ratio could be an indication. So can it be a blood test? Unfortunately, we always, so if you go to physical therapy or annual physical screening, people do take the blood test and do the ALT AST test, liver function test. And unfortunately, that is a global test of your entire body. So it's not from the pancreas. So it's no direct way of imaging by doing that. If you can take the blood from the pancreas, maybe there is a way of seeing that. So in fact, we're working with something called circulating DNA and circulating exosomes from the pancreas to see if it's actually holding true in that. So that's a whole other area. I do not have the data to talk about it. And we are wanting to work on that as well. And in fact, Shivanand is working on some of the NMR spectroscopy of exosome and platelet. That's a whole other area. Hopefully someday he'll talk about it. But thank you. We are not quite there yet, to be honest.
Great, thank you. Thank you so much.
Okay, we have one last question from Grace. Grace?
Thank you, Suri. Very nice, informative talk. I was wondering, you showed it takes 4 days to diagnose the lactate levels in mouse. How many days in human beings will it take to recognize the cancer is coming down?
Oh, that's a great question. We didn't do 4 days, just we're going to give, because our, we could have done it earlier, just to let you. The reason we did it on 4 days, because MD Anderson and most universities here are very strict animal protocol. So once we inject it, we can't just inject it again. So it's a part of the patient. But I can tell you on a completely different system, in fact, one of my graduate students who just graduated work on the same system on brain tumor. In fact, tumor, we saw this within a day that the lactic acid goes down in brain. So in fact, one of my lab, we published a paper. This is when I was at USC before I moved to MD Anderson. This was published, I think in 2012 at JACS, Journal of American Chemical Society, where we injected a hyperpolarized, in this case, succinic acid made by parahydrogen method in the liver of a mouse. And we can see within 10 minutes, the metabolism is altered. So what we find that hyperpolarization, essentially, the metabolic changes happens really fast. If it is supposed to happen, it will happen. If it doesn't happen, it never happens, even after like a day or so, even after that second. So that's what we're seeing, that metabolic changes precedes any anatomical change. And if you can actually capture that, and in fact, you're right that we should probably do that in human. In fact, that was a trial we are planning. But as you can understand, in United States, it's a very rigorous FDA review. And it is, I know our protocol is somehow stuck in animal and human IRB review. So hopefully someday it will be accepted and we can try this in human. So that was that clinical trial in prostate cancer. We will treat them with a drug called enzalutamide, which is the androgen therapy. And the idea is how early can we detect them? That was the experiment in clinical trial we planned, but it's still stuck in the regulatory chain right now. So hopefully we can able to do that some point, but hopefully it'll be soon. I do not have human data to answer your question. So I'm assuming it'll be really soon, within a day.
Okay, thank you, Pradeep. Now for want of time, I think we cannot continue with any more questions. If there are any questions, you can write to us. We'll communicate to Dr. Pradeep through email and you can get your answers. Okay, thank you, Pradeep. So thanks.
Feel free to email me if you have any questions.
Thanks both the speakers, Francesca and Pradeep today for your excellent presentations. And again, we'll meet next week. I'll show this slide of the next week, who are the speakers of next week once again. So these are the speakers of next week. Again, so I invite all of you for two more exciting talks at the same time next week, Tuesday. Thank you very much for all of you.
Thank you.
Thank you very much, both speakers. Wonderful talks.
Bye, everybody.
Bye, Francesca. Bye, Pradeep. Thank you so much.
Thank you very much.
Thank you. Thank you.